Good afternoon, everyone. Let's go ahead and get started. I'm Priya Verghese. I'm the Division Head of Pediatric Nephrology at Lurie Children's, and I'm really delighted to introduce our speaker for today, Dr. Matt Sampson. He has a strong East Coast rooting in terms of his education. So he did his med school in Virginia, did a MS in epidemiology at Duke. Did I mix those up, or is that right? MS at UPenn, and then went to CHOP for residency, fellowship, and got his first faculty position at Ann Arbor, Michigan, which was the first time he got introduced to our Midwest winters. He is currently an associate professor at Harvard, where he has his own genomics lab. He has multiple publications, an R award, all towards figuring out how to explain and help treat nephrotic syndrome through genomics. So he's considered the kidneyomics expert. And so it is my delight to invite Dr. Sampson to give today's Chicago KUH lecture.
Good afternoon. Thank you so much, Priya, for the introduction, and thanks to the Chicago KUH program for inviting me. When I was invited, it was an easy yes. I've been here multiple times over the past decade or two, and I have such great love for Chicago and for this health system, and gotten to meet faculty on the pediatric and adult side. It's always wonderful to meet y'all elsewhere, and it's good to be here. So thank you for your hospitality. So this is a brand new talk for me. I was inspired by coming here to put together some new ideas and thoughts and present some unpublished data from a couple of the folks in my lab. So I'm going to talk, so while I'm a big social media guy, it would be great if you didn't share most of this stuff. So we're going to talk about the value of genetics in improving the care of patients with nephrotic syndrome and the unique opportunities at this point in time for kidneyomics research. And then I'm going to talk about 3 projects that highlight the power of the EHR, population-based biobanks, that's really aiding in our arc of discovery to medicine. You might recognize that this is not the genetics of steroid-sensitive nephrotic syndrome. As I was putting together the talks for this week, I thought this would be a lot more fun to talk about. So if any of y'all are only here to learn about GWAS of SSNS, I hope you got a good lunch, and if you want to leave, feel free. So part 1. So I have been bugged since I was a first-year fellow, taking care of children who I could easily diagnose with nephrotic syndrome based on hypoalbuminemia, proteinuria, that we are very imprecise in our classification of patients. You know, if they respond to steroids and they're sensitive, they have a diagnosis of steroid-sensitive nephrotic syndrome. If they're resistant, SRNS. Or we do a biopsy in pediatrics, that might be later. Y'all in adult medicine, I think you do it a little earlier or at first. And if you have minimal changes, you have minimal change disease. And if you have focal areas of glomerular sclerosis, you have FSGS. These have been helpful classifications that have empowered us to do clinical trials and observational studies, but they really don't dissect or describe the precise molecular anatomy of the condition. And when I was doing this in nephrology at CHOP during training, my colleagues in HemOnc were not saying you just have blue cells in the blood syndrome or you have lump in your leg syndrome. They're saying, you know, you have an NMIC duplication, or you have a transversion, and, you know, cystic fibrosis was delta 506, and they're working on drugs. And I was very jealous, and I was annoyed, and I was frustrated. And so with the impetuousness of youth, I said, I want to get after this. And it's like 16 years later, no one told me I need to stop. And another bite of this frustration is this like circle of poisonous medications that we put all of our patients with nephrotic syndrome on. Sometimes, you know, these drugs go from Cytoxan to Cellcept, tacrolimus, rituximab. Sometimes they work about in achieving remission, but what they always do is have side effects. And this is not an unprincipled approach to go in this circle. It is principled, but, you know, I think we can do better. And we can take inspiration from our colleagues who've used genomics and precision medicine to improve the care of patients with sickle cell disease, beta thalassemia, cystic fibrosis, ulcerative colitis, asthma, to do the same thing for nephrotic syndrome. So lots of people are equally annoyed as I am, but I decided to move my annoyance towards genomic discovery, which I call kidneyomics. So why kidneyomics? I think if we understand the genetics of nephrotic syndrome, we can discover disease-causing or disease-contributing genetic variants. And this is our entree into doing molecular and epidemiologic work that's based on a genetic signature. So we can use genetics as a biomarker. So even without understanding mechanism, we can say individuals who harbor an 8ball1 high-risk genotype or a Mendelian form of nephrotic syndrome, they have a set of clinical characteristics that can help guide us. But they can also deliver novel insights regarding nephrotic syndrome biology. So some of my colleagues who were practicing in the 80s and 90s said at one point in time it was unclear what cell type was driving nephrotic syndrome. Apparently the mesangium was really hot at one point in time. But then through Mendelian discovery, the podocyte as the key cell driving disease came to the forefront. And moving forward, we've learned a lot about calcium channels and actin cytoskeletons and APOL1 pores through biology. And that biology is helping us to identify new targets for medicines. So we're not obligated as nephrologists to do all of these things, to go from, the pithy way of saying is mapping to mechanisms to medicines. Each of us doesn't have to do all of these things, but I'm pretty convinced that if I within my lab can work and identify associations and genetic drivers, that I could bring that to my colleagues at the bench or in biotech and pharma, and we could work together on therapeutic strategies. And importantly, I think there are a lot of good drugs that exist that have been tried for nephrotic syndrome, FSGS in the past, and some that are on the shelf that have never been thought of to be used. And sometimes these drugs have failed in trials because we are putting patients with FSGS, I don't know if the camera's on, but I'm doing air quotes for those who are just listening, FSGS or nephrotic syndrome, which is really a collection of 10, 20 different molecular forms of the condition. We're giving a drug that might work for a tenth of them, but so 90% of the people in a trial are failing, but 100% of the patients for whom have the genetic signature that drug would work would be effective. So in my opinion, we need to bring genetics more to the forefront. And ultimately, if we can do this, we can go from genomic discovery to genomic medicine, and we as practitioners can deliver genomically informed treatments and care towards treatments and cures. So that's been the fundamental aspect of my lab for improving the care of people with nephrotic syndrome through genomic discovery. What we do in our lab here at Boston Children's is, we aspire to continue to illuminate the genetic contributors of various forms of nephrotic syndrome. We want to make discoveries that improve the health of people with this condition. And importantly, we want to train the next generation of kidneyomics investigators. And I've been so lucky since I arrived in Boston in 2019 to train 5 Mass General Brigham adult nephrology fellows, 4 pediatric nephrology fellows from Boston Children's, and welcome many postdocs who are PhDs and MDs from around the US and world to engage in this kidneyomics work with me. And we're going to talk about some of the work done by them later. So I'm assuming not all of you are as obsessed with kidney genomics of nephrotic syndrome as I am. So I thought I would spend one slide talking about the background of what we have already. And this is a key point of my talk today, is that it's important that we continue to discover new genetic contributors or causes of nephrotic syndrome. But we have a lot of genetic information already, and it behooves us to try to make the best use of that genetic data while also working on discovery. So monogenic forms of nephrotic syndrome, they are due to rare variants that likely none of us in the room harbor, and in fact, in all of Chicago, there may be 1 or 2 people who harbor a rare variant. But when that variant is present, it breaks a gene, and the person develops nephrotic syndrome. And what we know is the genetic form of the clinical form of monogenic nephrotic syndrome is steroid-resistant nephrotic syndrome. Patients who respond to steroids, calcineurins, rituximab, almost never have a Mendelian form of the disease. And there are about 60 monogenic forms of nephrotic syndrome, monogenic nephrotic syndrome genes. And about maybe what we've known historically is maybe 10% of kids in the US have that form of nephrotic syndrome. So it's not common, but for each of those patients who have it, it matters. And if we think about the lab tests that we do in clinic or in the ER, whether it's viral panels, ANAs, ANCAs, 10%, we do a lot of tests that aren't 10% positive. In addition to monogenic forms of nephrotic syndrome, there's also APOL1 kidney disease. Now, APOL1 kidney disease are caused by common variants in the gene APOL1, and those variants arose from West Africa about 9,000 to 10,000 years ago. And they're almost every person who has APOL1 genetic risk variants are of West African ancestry or would self-describe themselves as black. There are about 5% of individuals who would self-describe themselves as Hispanic have a high-risk genotype. So 13% of people who would describe themselves as black, 13% of individuals have these APOL1 risk variants. They greatly increase the risk of FSGS. They modestly increase the risk of developing CKD. And probably whether you have Alport syndrome, IgA nephropathy, lupus, if you have the high-risk genotype, you do worse. So we have monogenic and APOL1 genes that we can use for inquiry. And one of the challenges, and one of the things I'm going to get to, because we're talking about population scale discoveries these days, most of these correlations and most of these discoveries have been found by studying relatively small numbers of patients in nephrotic syndrome cohorts. So it goes like this. Someone's really interested in nephrotic syndrome. They say, let's collect every single nephrotic syndrome patient that comes into clinic. Let's focus on those who we think have a genetic form of their disease. Let's sequence a couple of genes, and let's understand the correlations. Historically, that's been the way it's gone, and I'll talk more about that. But there's a lot to think about that have limitations, and we'll talk about that later. So one of the reasons I'm obsessed with figuring out the genetics of nephrotic syndrome is there's these 3 papers summarize a large body of work that shows that if a drug that you're developing or using has genetic support for its indication, it's more likely to be approved and it's more likely to be effective. So going from left to right, this was a recent editorial in Nature Reviews drug discovery that says in 2021, the FDA approved X number of drugs, and 2/3 of those drugs that were approved had a genetic support. This was a paper in Nature Genetics about 4 years ago that said that drugs that are supported by human genetic evidence have about a 3 times increased odds of moving past phase 1 and phase 2. And this was a recent follow-up by some of the same authors accepted earlier this year that says that they estimate the probability of success for drug mechanisms with genetic support is about 3 times higher from those without. So if we're thinking from a biotech perspective and a drug development perspective, or we're thinking about as academicians, we're going to devote time and energy to work on developing a drug or a therapeutic or mechanisms, I would want to work on a drug that had more chances of being successful than those that aren't. But again, we don't, one example of this is these genetically inspired treatments for APOL1 associated FSGS. So APOL1 was discovered in 2009, and in the past 15 years, there's been a race towards mechanisms and medicines, which has culminated in an inhibitor of APOL1 protein, a small molecule. New England Journal of Medicine earlier this year in a phase 2 study showed that each of these orange lines, the longer the line, the more proteinuria reduction there was. Each column is a person. That there's early evidence that a genomically inspired treatment for APOL1, inaxaplin, which blocks the pore, improves health. And Maze Therapeutics is another company that is making a genomically inspired treatment. And this should be super duper exciting for us because all of a sudden biotech and pharma is thinking about drugs for kidney disease and thinking about genetically inspired ones. So that's one motivation. But again, it's not just about, this is like the ultimate goal, right, to develop treatments and cures, but we can do a lot of good for our patients if we just have a better understanding of the clinical implications of a genetic signature. So even without targeted drugs, genetically informed care is better care. I had the opportunity to be part of a KDIGO and an NKF working group that talked about the value of genetics in CKD. And this is very small, and I have old eyes. That says, they really recommend people who are taking care of patients with CKD to think genetic. And, even without developing a drug, you can find, if you think genetics among your CKD patients, you might identify patients who do benefit from a specific therapeutic. Conditions amenable to non-specific renal protective strategies, Alport syndrome and ACE inhibitors, for instance. Avoidance of prolonged immunosuppressive therapies. We are very special to have kidney transplant as part of our armamentarium of treatment. So knowing the genetics could help us counsel individuals who are at risk for recurrence, as well as identifying conditions where a person should not be a kidney donor. And finally, a lot of our kidney conditions might benefit from extrarenal screening. So we should really be incorporating genetics into our care of patients. And I'm sure you guys are doing this, and I'd be interested in hearing. And I've talked to folks over time about what's being done here. And I have the unique perspective in Boston, being part of Boston Children's as well as the Brigham and Mass General, to see how we're all progressing in this path together. So that's the background. That's the motivation that's gotten me engaged in my head and my heart for the last 16 years on this project. And now I want to talk about why now in 2024, this is like a perfect time to work on doing genomic kidney omics research. And what I'll assert here is that less expensive technologies paired with more expansive cohorts are putting us in this exciting position to use EHR-linked biobanks to increase our understanding and utility of knowing genetic forms of nephrotic syndrome. So let me explain what I mean by that. So this is, if anyone has gone to a genetics talk or a sequencing talk, it's almost obligatory that, you have to put this picture in. And what this is, the x-axis is time and the y-axis is cost. Moore's law is coming from computing, which says every X number of years there's a massive decrease in cost of, an extra, processing power. And this orange line is the cost of sequencing that has decreased from about, $10 million to sequence one genome in 2001, and now in 2024 at the Broad Institute, where I work partially, that you can do a genome sequencing for $99 a sample. And not only can you do genome sequencing, which gets the DNA, which is basically the same in your eyeball and your ear and your elbow, but you can also get single-cell profile RNA sequencing, ATAC sequencing. These are molecular measures of gene expression and open chromatin that lets us know what genes are turned on in the podocyte versus the endothelial cell. You can generate proteomics data, metabolomics. You can do long-range sequencing. You can do single-cell. So we have the opportunity for a cost that is attainable for folks who are on R01s or U grants to sequence individuals more cheaply. And the cheaper you are, the more patients you can sequence. And when we do association studies and clinical, this is just a different form of like clinical research. The larger your sample size, the more power you have to detect smaller effect sizes, which could nonetheless be very clinically meaningful. You guys with me? Yeah? All right, good. Not only is sequencing cheaper, but now we have access to huge biobanks with existing genetic data. So this is from a recent review a couple of years ago, and these indicate biobanks that exist around the world. Looks like the closest one to us today is the Michigan Genomics Initiative, 55K. They're up to like 100,000 K. Got one at, what's here over here? BioView, that's at Vanderbilt, 120K. Here, there's about 2.2 million individuals who are part of research-based biobanks that for some, they're open access with minimal hurdles to get in, like the UK Biobank. Some of these others require a little bit more work. Some of this data is publicly available. This morning, Gal and I were on the computer looking up some of these biobank information, which is really easy for me because I do it all the time. I can't do anything that she does because she's really excellent at that. But we have the opportunity to study existing genetic data, but also generate genetic data from 2.2 million people. And what I'm going to argue and what I'm going to show you is that when we start studying the nephrotic syndrome genetic signatures in big biobanks and people who are not selected for having nephrotic syndrome, we get to learn all sorts of interesting things. The other value of EHR-based biobanks is that they collect indiscriminately all sorts of clinical data that and over time that gives us more opportunity to make associations. So why is this approach so exciting? I think I've been talking about my excitement constantly here. So these larger sample sizes, all of a sudden, I talked about rare Mendelian variants that might exist in 1 in 100,000 people, or maybe 1 in 50,000 people. All of a sudden, when you're working with a sample size of 2 million individuals, a rare genetic event becomes more detectable. Or if you're looking for a rare clinical condition like nephrotic syndrome, which might take years to build up a cohort, if you have 2.2 million people or 1 million people, these patients may be discoverable. And when you discover individuals who have a genotype first approach or a phenotype first approach, we can then relate this group of individuals to all sorts of phenotypes or genotypes. Secondly, by doing biobank-based research in relation to nephrotic syndrome, we're removing selection bias. So historically, because remember the Moore's law and the graph that I showed you like 2 seconds ago, which I'm sure you all remember, that unless you're like Dory from Finding Nemo, pediatric nephrology joke. Historically, genetic sequencing is so expensive that you couldn't just go and sequence every person that comes into the adult nephrology clinic or the pediatric nephrology clinic, because most people are not going to have a genetic cause, and you'll be wasting a lot of money. So historically, all the genotype, phenotype, and prevalence studies have been focused on patients who are recruited who are likely to have genetic disease or those with a clear form of nephrotic syndrome. And this has limited our ability to understand the impact of these genetic variants in the broader population or in new scenarios. So it's a very relatively new thing to think about Mendelian forms of disease in adults. But in the past couple of years, there was a couple of real fundamental papers, including one by Ali Garabi's group in the New England Journal of Medicine about 5-6 years ago, that this new scenario, like Mendelian disease in adults, it matters. And as I said, it's also exciting to me because the EHR has expansive and longitudinal phenotyping. So we're empowered to discover associations that we'd be blind to if we relied only on cross-sectional studies. For instance, what is the impact of a genetic risk on progression of EGFR decline or advancement to ESKD? Or what is the association of like hearing loss or eye findings over time or at one point in time? So if we had a limited amount of time or money, we had a clinical research coordinator who was trying to collect the key bits of information, we'd be limited compared to, hey, what if we can access the entire chart? So that's some of the background. And what I want to talk about here in part 3 is what happens when we take these phenotype or genotype first approaches to large cohorts. This is work being done by the fellows in my lab and research scientists in my lab. Most of this is unpublished. Some of it was at the ASN this year. So the first story I want to talk about is a phenotype first approach. And this is the story of a genetic modifier of APOL1-mediated FSGS that has the potential to guide care right now. And this is a joint effort with Simone Sanakerke's group at Columbia University, Dave Friedman and Martin Pollack at Beth Israel and Harvard Medical School. And this is being led by Michelle McNulty in my lab. So the background, we just talked about this. This is about APOL1 kidney disease. So high-risk APOL1 genotypes are associated with a 15 to 20 times increased odds of FSGS and increased odds of many other kidney diseases. 13% of all Black Americans have a high-risk genotype. And because this is not a Mendelian disease where genetics is destiny, there's incomplete penetrance. But what we now know is that of individuals who have a high-risk genotype, there's about a 20% lifetime risk of getting any kind of kidney disease. We also know that FSGS patients who have a high-risk genotype do worse. And when we flip the scenario on its head and we think about individuals who self-report as Black, who we see in our nephrology clinics who have FSGS, 70% of those Black Americans have FSGS due to APOL1 kidney disease, which implies if we come up with an APOL1 drug, we have the opportunity to help a huge percentage of this population who have FSGS. And APOL1 testing may also worsen racial disparities for kidney transplant. So if we're in a small group, I'd ask you to imagine and tell me what you think, but we're not, so I'll just tell you myself. So, there are already racial disparities in kidney transplant due to incompatibility with deceased donors and also challenges in living donor transplant. Now, individuals who have a family member who have a high-risk genotype who might want to donate, the high-risk genotype is comprised of 2 APOL1 risk alleles. So if a parent wants to donate, they at the least will have 1 risk allele, but they may be more likely to have 2 risk alleles as well. And there's this body of literature, and there's a whole longitudinal study, the Apollo study. Are y'all involved in Apollo here? Yes, I see a head nod. Are trying to figure out what it means to have an APOL1 high-risk genotype in terms of transplant. But right now, there are centers that are concerned that, do I have a picture of it? No. Okay. There are centers that are concerned. So I've had a situation where I had like a 16, 17-year-old brothers who were on dialysis, end-stage kidney disease. They both had APOL1 high-risk genotype, and their parents were in their 60s, wanted to donate a kidney to them. They've passed all the typical transplant screening, but they also had high-risk APOL1 genotype. And despite the fact that only 20% people only have a 20% lifetime risk of developing kidney disease, and despite the fact that the APOL1 kidney disease typically has an onset in the late teens, early 20s, these parents were were rejected to donate a kidney based on this APOL1 high-risk genotype. But what about this? What's the challenge? Given that only 20% of patients with this genotype develop kidney disease, can we somehow identify those high-risk individuals or high-risk kidneys who will never become APOL1 kidney disease patients or kidneys? So are there protective factors genetically that could help us look at a person with a high-risk genotype and say, you're never going to develop the kidney disease? This has a profound implication for people at risk of developing kidney disease and profound implication for to donors and recipients. So does this exist? Obviously, because I'm sharing this slide here at this talk, right? So in vitro work by Dave Friedman at Beth Israel in 2019 took in vitro studies and looked at APOL1 haplotypes, and they have a haplotype that had this protein called this exonic change that changed position 264 from an N to a K, and they found that people have the high-risk APOL1 genotype who, in addition, had this amino acid change. It was protective. Basically, you gave interferon, and if you have the high-risk APOL1 genotype without the N264K, those cells died, but these cells didn't die. And then there's population-based work that showed that 5% of people with a high-risk genotype have at least one G2 allele have P-N264K. And for the people, if you want to get really deep into this N264K occurs on the same haplotype as G2, not G1. So we're really looking for those 5% individuals who have one G2 and N264K. Now, if we were going to do a prospective study, we'd have to think about 5% of the 13%, so 5% chance of this genotype happening in black individuals. 13% of the US population is black, and 13% of those black individuals have the high-risk genotype. So basically around 8 in 10,000 black Americans will have that. It would take a long time to recruit and find those individuals. But because we have access to biobanks, we were able to do this very quickly on existing data. So we had a hypothesis that we want to follow up on Dave's work in cells, and we hypothesized that the high-risk genotype modifies the risk of FSGS amongst those with a high-risk genotype. So what we did is we looked in a Columbia-based biobank, and then we looked in 3 other biobanks, Emerge, Regards, which is a study of cardiovascular disease in individuals in the South, and 1 other. And what we started is by finding individuals who are high risk, and these 3 genotypes represent high risk. We identified genetically matched controls. This is another value of biobank-based research, because anytime we do these genetic studies, we have to do case control, and it's not enough to say you have kidney disease and you don't have kidney disease. Because if you put if there are lots of white people in the kidney disease and lots of East Asian people in the no kidney disease group, the genetic differences of the association study would just be the genetic markers that differentiate those global populations. So we have to identify genetically matched controls, and it's only these biobanks that have a huge amount of patients that allow us to do that. And then all we did is basically quantify the relative frequency of this variant in cases and controls. So in cohort 1, which was FSGS only, we had 528 cases. If you think about how many people you'd have to screen to get up to 528, hundreds and hundreds of thousands. We had 2,606 controls. And what we found is that those individuals with the high-risk genotype of N264K had an odds ratio of 0.08. So they were highly protected against getting FSGS. Not only that, but if there was no individual that existed in these biobanks, of these 2,606, 528, no one who was G2 plus APOL N264K had FSGS. So what this suggests is that if you have a high-risk person, okay, not patient, person, if you are a high-risk individual who also harbors the N264K genotype, your risk of getting FSGS is no different than the regular population. And of course, we had to replicate our work, and we also want to expand the phenotype, because does this benefit just for FSGS? And the answer is no. So we then went to this independent cohorts of individuals with stage 3 CKD as well as ESKD, did the same thing, and found a replication. So not only does the high-risk genotype protect against getting FSGS, but the high-risk genotype plus N264K also protects against CKD in general. And around the exact same time, a study led out of Vanderbilt by Alex Bick and Adriana Hung that took advantage of the Million Veteran Program, the MVP program, which has recruited like 500,000 veterans in the United States. The Vanderbilt BioView and the NIH All of Us study found the same thing. They found that in terms of CKD, the N264K variant in context of a high-risk genotype has a 0.43 odds of developing CKD, and then the same thing, they had about a 0.2 odds of developing ESKD. So now we have known APOL1 genetic risk factor. We now have a protective variant, and we found in 2 different studies in 3 different cohorts empowered by biobanks that suggest that this variant is protective, and and I would assert, and there's a follow-up abstract of the ASN that supported this, that this matters. So Rashid Badagashian led a paper that was in glomerular disease this year where we talked about this. I'd refer you all to that for a bigger discussion. So here are the clinical implications. So it used to be that the risk genotypes, that there are 5 different haplotypes for APOL1 now. There's the G0 haplotype, G0 with M1, G1, G2, G2, M1. Individuals who have the G2 M1 genotype should be reclassified as a non-high-risk genotype. And what that also means is that if we go back to those parents with that 16, 17-year-old, if those individuals in their 50s and 60s were healthy and they had the high-risk genotype but they had the M1 allele, in my opinion, and we need to do some more research, but I think a lot of research has been done, we should think about these individuals having a low risk of developing APOL1-mediated kidney disease. This is also, if you change this from a clinical classification to a drug target, if we could basically make a drug called APOL1 N264K, figure out the biology, it might be a new target for novel therapy for this condition. Basically, we think that it is making the pore, the APOL1 pore that punches holes in podocytes, not be as poisonous. And finally, if we're doing clinical trials for like that inaxaplin or the MAZE study or any other sort of ASO study, if we have patients, so remember, 13% of black individuals in the United States have a high-risk genotype, but individuals are allowed to have kidney disease not due to that genotype. So if someone says, oh, they have a high-risk genotype, we don't need to do any additional studies, I think what we really need to do is understand if those folks have N264K, because if they do have N264K, we need to be diligent about figuring out other causes of their kidney condition. And furthermore, when we're testing drugs, we do not want to use an APOL1 drug in a clinical trial against patients who are incorrectly attributed to have APOL1 kidney disease. That should give us more precision and increase our potential for being effective, and decrease the side effects. All right, so there was an ASN abstract. We're working on a follow-up paper that expands this quite a bit. I have a disclosure. I'm on the scientific advisory board for Natera, but that's not really affecting any of this work. I'm not advertising the Natera panel, but I know that Natera and other folks are having discussions about whether we're going to report out M1 on the sequencing studies in the future. Okay, so that was one study. The second study I want to talk about is another phenotype first study, and this was led by Jane Witt-Wong-Boonson, who's an instructor in medicine at HMS, a staff nephrologist at the Brigham. He also does a lot of his clinical work back in Bangkok. He moved back there, and he's been in my lab as well. And what he reported on is the prevalence and clinical correlates of genetic forms of FSGS and MCD in the Mass General Brigham Biobank. So the rationale is that few genetic studies of nephrotic syndrome have been conducted in adults. And those which do exist are almost exclusively a study of Mendelian nephrotic syndrome in adults or a study of APOL1 high-risk genotype in adults. And almost exclusively, these sequencing studies are, again, from patients from nephrotic syndrome research cohort or someone going into a nephrology clinic and saying, there's a research study available, will you recruit? So there's a bit of a gap in terms of understanding from an all-comers perspective what these genotypes, how prevalent they are in the population, what they mean. So the question that Jane really wanted to attack is, what do we learn by studying adults with FSGS and minimal change who happen to be enrolled in EHR Biobank? And they really happened to be enrolled. No one selected them. They just were at Mass General. They got a message in their chart, or they were approached by a study coordinator at the blood draw station or elsewhere saying, do you want to be in a study? Okay. I'll also note that as a PI who's always thinking about projects for my team, that the available of existing data like this, the barrier for entry into this is just enthusiasm, industriousness of a trainee, and this is not a huge investment in money. And so these are ideal projects to get folks started. So that's what we want to do, but the question we asked ourselves is, can we find nephrotic syndrome patients via the EHR alone? Jane Witt's a really hard worker, but I couldn't have had him go through 112,000 charts by himself. So we had to figure out, yeah, exactly, Rhodes knows what I'm talking about. So we had to come up with a strategy to identify rare diseases in an EHR cohort. So this is what we did. The first thing Jane Witt did is we tried to assess what would be in a strategy that would most accurately identify nephrotic syndrome patients in MGBB, accurate and efficient. So accurate means maximizing sensitivity and specificity. Efficient means, don't spend 4 years going through these charts. So the challenge was we started with 130,000 participants, and we used three strategies. The ICD-10 strategy was basically look for anyone with an N04 code. This was a strategy that I thought, my thought was it would maximize sensitivity. There might be a lot of patients who are coded that way who don't have the disease, but this is a rare disease, and I'd rather be more sensitive than specific. Another one we worked on is a computable phenotype that was developed by folks at University of Michigan. They used a combination of 8 inclusion codes and 37 exclusion codes from ICD-9 and ICD-10. And we thought that'd be a very precise, accurate, and specific way of identifying patients. And the third one that Jane Witt came up with is, he said, hey, why don't we look at the 112,000 individuals who have a pathology report that also includes like blood tests and other sorts of pathology. And let's just like search for the words glomerular, glomeruli, glomerulus, mesangium, and podocyte, and then see what we got and go from there. So in doing this, we found 558 participants using the ICD-10, 86 participants using the computable phenotype, and 2,014 participants who had the path report, who were qualified on the path report. And then, so this was quick, but then the adjudication had to occur. So Jane then undertook a Herculean effort on chart review. And of these 558 participants with a nephrotic syndrome code, 35 actually had no kidney disease, which wasn't surprising to me. There are a lot of patients that I see who get coded for billing for nephrotic syndrome, and there was just concern for. Out of this 86 patients with a computable phenotype, remember, out of 130,000, only 86 came up with a computable phenotype. 29 of those individuals didn't have kidney disease. And of the 2,014 people with the path report with these words, 302 had no kidney disease. So, and then of those 2,014, 543 had allograft pathology, and 803 only had native kidney pathology. So then we underwent chart review of those people who remained. So there are about 1,800 patients with kidney disease. And then only 618 of those individuals had nephrotic syndrome. A lot of those patients who had kidney disease but not nephrotic syndrome had things like post-strep or IgA nephropathy or MPGN, et cetera. And then we had a cohort of 618 individuals, adults, with really well-adjudicated FSGS and minimal change. And those that happen to have exome sequencing data was only about 70,000, only 70,000 people in Mass General Brigham Biobank have exome sequencing data, but 319 of our nephrotic syndrome patients did. So again, through biobank-based discovery for a rare disease and some time on task, we had a cohort that's pretty expansive. And because it's exome sequencing data, we had both the ability to adjudicate Mendelian forms of disease as well as APOL1-associated kidney disease. So the first thing we did, because we're working on submitting this, first thing we're talking about is the different methods of screening, because I could anticipate other folks wanting to do this. This is an upset plot. This is the ICD-10 strategy, computable phenotype strategy, and the pathology strategy. This is the sample size that was ultimately grabbed that were positive based on all three approaches. And you could see the computable phenotype did not do very well in identifying individuals, whereas the pathology identified a lot of them. And what you found here is the individuals identified by pathology alone represented more than half our sample size of 623, whereas the computable phenotype alone, only 3 individuals. So that would suggest that if I only could pick one strategy, I would do the pathology report alone. And with natural language processing and AI, I think moving forward it might be a lot easier to identify true positives of that ultimate group of 2,000 that Jane would have. So what did we find? So ultimately, we found that 18% of Mass General Brigham Biobank patients with FSGS or minimal change, these are adults, have a genetic form of their condition. So there were 319 individuals with a nephrotic syndrome spectrum of disease. Of those, 67 had positive variants, which we defined as being pathogenic or likely pathogenic using ACMG guidelines. But of those 67, there were 12 who had the variants that were not related, that we could not attribute to their kidney disease. So they were positive, but and we had a limited number of genes that we were checking. But there's a gene like MEFV, which is associated with like fibrillary nephropathy, glomerulonephritis, that we couldn't really attribute to this condition, so we kicked those out. So of those 55 that came in, 24 individuals were APOL1 high risk, 10%, and 31 individuals had Mendelian variants, 8%. Again, these are adults. These are the genes that were implicated, and those of y'all who are in the know won't be surprised that the most common variants seen were in the COL4A genes. INF2 has a lot of face validity as well. But you also see that there are genes that phenocopy FSGS. So UMOD is like a gene of the tubular interstitium, but you can get secondary FSGS from that as well. And then there are a couple of others in here as well. Tuberous sclerosis gene, IFT140, was the cystic disease gene. But quote unquote, it's a cystic disease gene. Like, is it really a cystic disease gene if through chart review patients were diagnosed with nephrotic syndrome, didn't have cysts on ultrasound, and had a biopsy with FSGS? And breaking that down a little bit more, 38% of these patients basically had a collagenopathy. 13% had an abnormality in the complement cascade, and then other genes represent 48.4. This again argues that you can't really do targeted genotyping these days for one or two genes if you're really trying to make the maximum diagnosis. These panels are more effective. So a lot of times studies stop here, especially when you're working on like cohort studies, but we had the EHR available to us, so we could do a lot more. So what I would say is there are some arguably unexpected genotype histology correlations. So Jamin went back and he found that a substantial portion of individuals with genetic monogenic FSGS and APOL1 FSGS were classified as secondary FSGS on biopsy review. So 18 of 22 Mendelian cases were classified as secondary FSGS, and 100% of those with pathogenic variants in complement genes also were called secondary FSGS, and about half of the APOL1 FSGS were called secondary FSGS. So in talking to our pathology friends and my adult nephrology friends, in research, a lot of times patients who are determined to have secondary FSGS are not thought to have genetic forms of disease and are kicked out of genetic studies. So this is suggesting that that's probably not a good idea. There was also 10 patients with COL4A pathogenic variants who also had electron microscopy. And only 20% of those individuals had features of a collagenopathy, like a thin basement membrane. These were all reviewed by the Mass General Brigham pathologist clinically, Helmut Reinke and the squad over there, Astrid Wines. And so again, this just further supports the idea that COL4A pathogenic variants are not always going to show up as like an Alport phenotype or a thin basement membrane. And then we looked at ESKD in patients as a function of their genetic signature. So because we had longitudinal data, we could do this over time, and we linked these patients with USRDS. And what we found is that in this multivariable model that adjusted for all the things you'd want me to adjust for, that in the Mendelian group and the APOL1 group, there was increased odds of going to end-stage kidney disease. The Mendelian form, individuals with Mendelian form of disease had a 3 times increased odds, APOL1 6.5 increased odds. And again, there's a similar pattern when we used time to event hazard ratio approaches. So the conclusions and potential implications. So we've devised a way to identify patients with a rare disease in hospital system-wide EHRs, and we need to get more efficient. I think AI and NLP will help to do that as well. And I'm really interested in knowing if this phenotyping strategy is portable to other health system EHRs. We have independent work on the pediatric side doing this as well. But wouldn't it be great if we had the same approach that could work in Chicago, in LA, in Boston, et cetera, that we could grab all these patients and feel really good that we've built a synthetic APOL nephrotic syndrome cohort. These patients don't look exactly like they do in research cohorts. Patients with secondary FSGS should not be excluded from genetic testing on a clinical side as well. And I think in the future, what we're going to be doing is there are more genetic signatures that I haven't talked about yet right now, like genetic forms of steroid-sensitive nephrotic syndrome. And there's also polygenic risk scores for chronic kidney disease, venous thromboemboli, hypertension, et cetera. So I think ultimately we are going to get a more holistic genetic profile of the patients that come in, and it's going to be clinically meaningful. Okay. So my last story is a Genotype First project, and this is led by Adrian Banerjee, who's a senior pediatric nephrologist here at Boston Children's. He's on the job market, and he presented this at ASN, and his story was now Genotype First. And what he was interested in are what are the clinical implications for people carrying single pathogenic nephrin variants or carriers. We have learned over time that carriers are not always benign. We've thought about this in terms of a lot of X-linked conditions where women have one copy of a collagen 4A5 gene, other conditions. So we wondered if the nephrin variants are associated with a problem when you're a carrier. So here's the background. So nephrin, NPHS1 encodes nephrin, which is an integral component of the slit diaphragm. And we know that Mendelian forms of nephrotic syndrome due to nephrin are due to biallelic pathogenic variants. So you have to have, it's recessive. You have one copy on your maternal chromosome and one on your paternal chromosome. And when these biallelic pathogenic variants occur like this, you often have congenital nephrotic syndrome for these loss of function variants. And sometimes if you don't have congenital nephrotic syndrome, if you have missense variants on both sides, you may get early onset nephrotic syndrome. And NPHS1 is a monogenic form of nephrotic syndrome was discovered in Finland due to founder effects. So Finland is cold and it's hard to get to. So only a small group of people showed up in Finland from Western Europe, Northern Europe a long time ago. And one of those individuals happened to have a pathogenic loss of function variant in nephrin called Fin major. And those folks procreated, and that rose to prominence. So let me tell you about the Finnish type of congenital nephrotic syndrome. Here's a map of nephrin. So these are all the exons, the dashes, and then we have fin major and fin minor are the names of the two most frequent mutations that lead to 90% of the congenital nephrotic syndrome cases in Finland. So these variants are fin major. This is the most important one. This is a frame shift variant, and fin minor is a stop gain variant. Now look at the minor allele frequency that exists in the Finnish population. 9 in 1,000 Finnish individuals have fin major, whereas in a worldwide population from the NOMAD study, where we've aggregated millions of genomes, only 2 in 100,000 people have these variants. Same thing with Finland, fin minor, an order of magnitude. So we have to go to a place like Finland to find enough people with this variant to do this kind of study. Well, I didn't go to Finland to do this because we had FinGen, okay? FinGen is a public-private partnership that enhances genetic discovery with a genetically unique population, the Finnish population. So the Finnish population is about 5 million individuals, and FinGen has already collected genotype data paired to health registries with over 50 years of record for 10% of the Finnish population. And they've newly released laboratory data as well. And historically, people in Finland have studied fin major quite a bit, and there's never been an implication of fin major heterozygous or fin major carriers as associated with disease. So the study design that Adrian undertook was to test the hypothesis that people with a single fin major allele have an increased risk of kidney disease compared to patients without NPHS1 variants. And I underline kidney disease and not necessarily nephrotic syndrome because we didn't hypothesize that a carrier might have nephrotic syndrome. They might, but we also wondered, would there be more proteinuria, would there be lower EGFR, et cetera? Much like sometimes women who have one copy of a COL4A pathogenic variant won't necessarily have Alport syndrome. They may, but they might have long-term hematuria. So we went to FinGen, which had 520 eligible participants. We excluded individuals who had other NPHS1 variants, as well as who were fin major homozygotes. Because if they're fin major homozygotes, they have congenital nephrotic syndrome by definition, and we didn't want to study that. So this was left with 519,000 FinGen participants, and because of the founder effect, almost 9,000 Finns have a single copy of fin major, and 510 individuals don't have fin major or fin minor. And this was a demographic table showing that essentially there's no real difference in proportion of male and female, no difference in age, and really no difference in percentage of individuals who are deceased. So as we think about robust epidemiologic models, there's no difference in these basic demographic characteristics. So what did we find? What we found is that there's actually an increased odds of nephrotic syndrome and ESKD in fin major heterozygotes. So this is our table. So I'll go row by row. So once you exclude patients with congenital nephrotic syndrome in FinGen, 28 fin major heterozygotes have nephrotic syndrome, and 838 have nephrotic syndrome who don't have a nephrin variant. The odds ratio is about 2, and the p-value is quite significant. When you adjust for things you'd want me to adjust for, it remains significant. In terms of ESKD, similar odds ratio. So carriers had a 1.7 times increased odds of going into ESKD, which maintained itself when adjusted. Now, one thing that we see here is that we did include patients with congenital nephrotic syndrome, even though there were 8 individuals who had congenital nephrotic syndrome and 1 who were carriers. Now, we could have kept them in, and I could have said, these patients are carriers, they don't have 2 copies, but my supposition is more conservative to say, for some reason we missed the second allele, or they have a rare allele, and it's not fair to be biasing away from the null to include these individuals, and it would be more fair and robust to exclude those individuals, even if a proportion of those 8 are true carriers with congenital nephrotic syndrome. So when we excluded them, the effect size was the same. The odds ratio was about the same, but the p-value, because we just had less events, was 0.14. I got my master's in clin-epi, and one of the things they hammered in, Harv Feldman, the folks at Penn, was like, pay attention not just to the p-value, but the effect size. So one possibility is as we increase our sample size under evaluation, this p-value will become more significant, but it is going in the same direction in terms of effect size. But the ESKD did persist. So what we can say right this second is that it seems that Finnish individuals who harbor one copy of Fin Major have about a 60% increased odds of developing ESKD. And these individuals don't have nephrotic syndrome. These are individuals who don't have nephrotic syndrome. These are people who are walking around who have ESKD, and we don't exactly know why they do. But they have multiple ESKD diagnoses in their chart. So the next steps is we want to do carrier associations with proteinuria and CKD stage by EGFR. We have data available over 7 years. We are going to restrict it to patients over 18. 60% of the individuals have at least 2 creatinine measurements, and the median is probably about 9 or 10. And 25% of individuals have UACR data available. So we think we're going to be quite powered to understand ESKD might be the most extreme phenotype, but are these individuals have a higher amount of albuminuria, et cetera. So we also want to extend this analysis, again, taking advantage of biobanks. We're working with Alex Cheng at Geisinger, who has the Geisinger Health System through the MyCode cohort has exome sequencing data on 200,000 individuals. We're not going to find a lot of Fin Major folks there, but we are going to try to identify people at Geisinger who have PLP variants in nephrin. And we're going to hypothesize that carriers of any pathogenic nephrin allele will have a higher odds. And that will provide more face validity of what we're finding. And again, these have clinical implications, and we need to think about that. And replication will be better. So, and then because we have access to all of us, the UK Biobank, MyCode, Mass Journal, our senior fellow, Adrian, will end up being able to study in a sample size of 1.5 million people. We're going to be able to identify new associations and feel good about our precision estimates. So the conclusions of this part of the story and the potential implications is, like I said, increased odds of ESKD. And these odds are still high when excluding congenital nephrotic syndrome. And the signal, so what we're saying is of all these individuals, this is not a bunch of Mendelian nephrotic syndrome patients where we're missing the second allele. I think there's a functional impact of losing half of your ability to make nephrin in your slit diaphragm. Are we going to be witnessing the extinction of the idea of nephrin carriers? Are carriers actually individuals who are affected? And will this have any impact on eligibility for being a kidney donor? So this is the part that feels really heavy to me and really motivates me to try to be, and our team, to be as rigorous and robust as possible, because we want to be very sure that to the best of our ability, we are accurate when will report that Finnish individuals who have a single copy, of which there's a large number, may have an increased odds of kidney disease. And then we need to pair this with genomic literacy and communicators who can discuss the absolute risk and what 1.6 times increased odds mean. So most parents, even if you tell them they're going to have a 60% increased odds, will give a kidney no matter what. And then we have to say 60% mean you're going to go from like 1 in 1,000 chance of CKD to 6 in 1,000. So we need to be able to communicate that very accurately. Same thing with APOL1. So, and we can extend this and say, does this matter for other heterozygous carriers, other carriers of recessive genes, POTISIN, LAM2, and because we have these biobanks, the rare genotypes become more common, and we have access to all sorts of phenotypes to allow us to look at things like proteinuria, et cetera. So this is my last slide. So biobank-based genomic research linked with clinical phenotypes has greatly improved our understanding of a variety of human diseases. And finally, we're at the point where we can do this for nephrotic syndrome research. And really having an integrated team made up of clinicians, data scientists, geneticists, and epidemiologists is really the way that we're making the most of this opportunity. And again, when your sample size is greater than 1 million or greater than 100,000, like you saw with MGB, the rare case and the rare genotype can become common enough. And diverse and longitudinal clinical data give us a chance to discover the unexpected. So with that, I'd like to thank our team. This was our Thanksgiving lunch the other day. We missed a lot of folks. Adrian and Jane Witt and Michelle did the bulk of this work. Alex Cheng, the Mass General Brigham Biobank, and the other people here were very supportive. And left a couple minutes for questions, so I'd be happy to have questions, comments, etc. Thank you so much.